My name is Gustav. I'm from the University of Florida, where I'm at the Florida Museum of Natural History, and I study marine invertebrates on tropical reefs. Marine invertebrates are basically marine animals. Life originated in the ocean, as did animal life, so the vast majority of animal diversity is in the sea, and all marine animals basically are invertebrates except for fishes and a few other marine vertebrates like marine mammals and reptiles. The diversity of marine life in the Pacific Ocean has a gradient where it's very rich toward the west and it decreases toward the east. So Moria lies fairly well along this track toward the east, so compared to islands like Fiji or New Guinea, it's relatively poor in diversity. Having said that, it's still immensely diverse. And one reason why biocode can be done in a place like Moria is because it's not so overwhelmingly diverse like, say, Fiji or New Guinea would be. And how many species there are, really, we have no clue. And for the really tiny things, we will not have a clue for a long time. For the bigger things, we're, really large marine invertebrates, which by marine invertebrate standards are about a quarter inch or a centimeter or more. You're talking thousands of species, and once you get down into the millimeter and submillimeter size range, you're talking tens of thousands of species. And I think this is our first year in this new biocode program, and by the end of it, I hope we'll have 2,000 species documented. In most days, there will be one or more field parties going out for collecting invertebrates, and the people participating in the field parties generally include some very experienced collectors who know the habits and habitats of the organisms they are after, so they can find them and they know how to capture them. A little bit of the diversity is captured just simply by hand. You look around and you take by hand what is out there. But because most of it is really tiny, what we do is mass sampling, which is you bring back a part of the world they live in and try to extract them by various means. So one method, for example, that we use quite extensively is to take a brush and brush the surface of the undersides of rocks, for example, into a net. And the brushings include all the little tiny critters in the 1 or 2 millimeter size range, plus and minus, that live attached to the rock that you would have a hard time even seeing in the field, and second, you'd have a hard time picking them up. And then when we come back, we have a bag full of this scuzz that we put under a microscope and pick out the animals out of that. So once you're back, the first thing that happens is you stabilize your catch, because they've been coming back in a vessel, they heat it up, they may not be as oxygenated, so we lay them out in dishes in the lab so they do well. They are then able to last for hours or days or weeks, depending on the type of organism and how far you need them to last live in the lab. And you put a station label with them so you remember where they came from. And this station label corresponds to a data entry in our field database program. And then you pick out the organisms into what we call morphospecies. And morphospecies are organisms that you think by your eye represent a single species, and you may or may not be correct about that. Sometimes different color morphs may be separated as different species when they're one, and conversely, often things that you think are one thing may turn out to be more than one. But it's a pretty good approximation, and for somebody who knows a particular group, they're usually 98% of the time correct in separating things into these morphospecies units. Once you separate them into morphospecies, we take several examples of each morphospecies and we take photographs of them. So we have live appearance recorded by a photo, which is very useful for later identification of those organisms, because you can identify a crab by the fact that it has red dots on a green background, or you can count the number of hairs on its legs, and it's much easier to look for the red dots on the green background than count hairs. So but a lot of this information doesn't exist because traditionally taxonomists received preserved material where the color is gone, so they ended up relying on the hairs. So a lot of the products from our project will be these huge photo documented inventories of the marine life here. And just this trip, we've been going for about 2.5 weeks. We are now over 15,000 photographs. Then once you photograph the animal, you then take a tissue sample from that animal, and that tissue sample will have DNA extracted from it, and a small portion of that DNA will be amplified by the polymerase chain reaction and sequenced. And in most animals, if you take a 600 or 700 base pair or letter sequence from mitochondrial gene regions, that's enough to tell that species from every other species in the world. And that's sort of the idea of genetic barcoding. It's not foolproof, but it's an extremely useful tool. And of course, you can use additional molecular methods to further characterize these organisms later as needed. So on the one hand, then you have a photograph, then you have a tissue sample, which will allow the genetic characterization of that individual. And then finally, the rest of the individual, which is the basic body, gets preserved and deposited in a permanent institution and identified by traditional taxonomic means. So that will allow us to put a name on it in many cases, most cases, I hope. So we will know what we're dealing with. I think the biggest challenge is that for all of us, this is a very intense and complex task. I don't know of any project that has undertaken a marine inventory, characterization of biodiversity in as broad a manner as we are attempting, that is across the board, across all of the marine animal phyla. And this means that we have to bring in specialists that know unusual groups, and sometimes the most diverse groups have the least number of people working on them in the world. This is sort of invertebrate bias. To give you an idea, there are probably 6,000 fish in the Pacific Ocean in reefs and maybe less. And there are probably tens of thousands of copepods. We could have found, literally dozens, if not hundreds of fish biologists to come in to do the work, and that actually has already been done. If you want to work the copepods up, you have 2 or 3 people in the world to pick from. We managed to get a Russian guy to come in and do one group of copepods, still leaves a couple of other copepods uncovered. But finding people to work these bizarre and really understudied groups is challenging. Second, we have a pretty good idea of the medium-sized invertebrates, sort of the 1/4 inch or 1 centimeter size range and up, as to how diverse those are. So we can plan, okay, we need somebody who knows crabs, we need somebody who knows echinoderms. But when it comes to the really small stuff, we don't know what to expect. And one of the things this year we tried is to get a handle on the relative diversity of the little things. So for example, among the small crustaceans, we expected isopods and amphipods, which is probably Greek to most people, but they're 2 little groups of crustaceans that are in the millimeter size range, to be both very diverse here. And we were planning to bring in an amphipod person and an isopod person next year. Well, it turned out the isopods are not that diverse, but the amphipods are mind-bogglingly diverse. So I think we're going to focus on amphipods, and the isopods will get done on the sideline. We'll get them done, but we won't have a specialized person devoted to that. So choosing your team is challenging. And then, of course, with so many people here and only so much facilities available to work with, the number of microscopes, the cameras, the bench space, everybody's, well, not fighting over it, but it's certainly an intensely used facility, let's put it that way. So that has been quite challenging. And then, there is the challenges in the molecular lab of simple things like reliable power supply for some of these high-tech equipment that people are used to running in the States, not on a remote island where the power supply is basically for light bulbs. And if you try to run a robotic extractor on that, and there's a big power spike, you're going to have problems. So there are many challenges, but we have a very enthusiastic group, and they're very happy to deal with the challenges and have a good spirit about it. I think the most exciting is just all the new stuff we're seeing. And my kids always ask me, what was the most interesting animal you found? And I can't say. I mean, we've seen so much cool stuff, and I think to me the most exciting thing, and for probably most people downstairs would agree, is that we have people who know different groups in different ways, and they will recognize what's exciting in their group, and then they'll scream out saying, look what I found. And invariably, whatever they are looking at is extremely unusual. I mean, you're dealing with animals with extra eyes, you're dealing with animals that change color, you're dealing with animals that live like hermit crabs, even though they're not hermit crabs, and there's just an unending supply of biological surprises and many new taxa, species, genera, that sort of thing. So there's just a constant flow of really exciting discoveries being made, and that really keeps a wonderful atmosphere downstairs of excitement and exploration.